Hello, everyone, and welcome to our latest webinar. My name is Joseph Ferra, Microfluidic Application Specialist at Fluigent, and today I'm joined by my colleague, Maya Baye, our Customer Support Engineer. We are very happy to have you with us for this webinar entitled Advancing Microfluidics Through Automation. In today's session, we will review the evolution of the liquid handling technologies from the earliest manual methods to modern automated microfluidic platforms. We will discuss their limitation at the microscale and how pressure-based flow control and automation has transformed workflows from organ-on-chip and advanced biological applications. We will end up with a Q&A session, so please send us your questions or any remarks that you have throughout the webinar, and we will address them at the end with the proper sessions that will be dedicated to. So don't hesitate to ask your questions throughout the Q&A panel at the bottom of the Zoom configurations. So for today's webinar, the guidelines will be, so first we'll start with the introduction and context from manual pipetting to microscale flow control. Afterwards, we will discuss three case studies: the recirculation for organ-on-chip applications, the second one, a droplet and microfluidics, and the third one, automated perfusion approach for spatial omics. And at the end, the Q&A session. So let's start a little bit with history about this field. So historically, as shocking as can be, mouse pipetting was the standard approach for transferring liquids in the laboratories. Operators would draw fluids directly by suction, which was not only highly unsafe, but also completely unreliable. So precision was essentially non-existent, and contamination risk were high. That's why we have come a long way since mouse pipetting, moving towards tools that provide both safety and reproducibility. This evolution represents the first steps of the long journey towards standardized and controlled laboratory techniques that we rely on today. One of the evolutions that was made, it was the Pasteur pipette, which was one of the first tools designed to improve safety and sterility in the lab. By using a thin glass tube and a separate rubber bulb, scientists could avoid direct contact with fluids, reducing the risk of contamination and accidental exposure. This simple tool enabled afterwards early microbiology experiments, such as transferring culture, inoculating media, and handling small volumes of biological fluids safely. It marked also a crucial steps towards separating the operator from the sample and led the foundation of modern volumetric pipetting techniques. In the 1950s, the invention of piston-driven micropipette transformed the lab workflows. These devices used a spring-loaded piston and an air cushion to aspirate precise volumes with interchangeable tips to improve sterility. So adjustable volume pipette allowed the researcher to accurately transfer microliter scale volumes, a capability essential for, especially for example, molecular biology, clinical assays, and let's say biochemistry experiments. So the standardization introduced by these pipettes enabled repeatable and reliable experiments compared to the past across labs worldwide, and it was adopted by the majorities of the labs around the world. Afterwards, by the 1980s, regulatory bodies officially banned mouse pipetting due to the safety concern, as you can imagine, and mechanical pipettes became mandatory in all lab settings. So this transition improves both safety and reproducibility, and modern pipette, whether fixed or adjustable, provided now precision and ergonomics, which enables scientists to handle very small volume consistently compared to the past. So this shift represented a broader trend in labs automation and standardization that continue to evolve today. However, despite all these these advances, the traditional pipettes are still not suitable for microfluidics and microscale, because like they cannot provide continuous stable flows at the sub-microliter scale. So manual evaporation still introducing variability in volume, timing, shear forces, making them impractical for the applications such as the cell experience fluid flow, or in organ-on-chip, the droplet generation, where all these applications we tend to search for precision and reproducibility. As you can see, for example, in the simulation of the pipette tip, there's that we can like prove there's known homogeneous velocity distribution and a high shear strain at the tip of the wall, which can produce uncontrolled fluid injection as the flow will be chaotic. And in this case, you will have high flow rates, high shear stress, and uneven injections, which can lead to sample damage and errors from intra-operators and inter-operators experience. So that's why there was several options to overcome these limitations. We'll focus on three main technologies that are commonly used in microfluidics. So the first one is the syringe pumps, which deliver precise volumes, but can generate pulsatile flow and have a slow response time. As you can see in the image, like it's based on a mechanical force, it's based on the piston that we are pushing. So like we'll have some errors or like low response time and a lot of pulsations in the flow, which can also affect the experiments that we are doing in microscale. The second option is the peristaltic pump, which usually support the circulation of the fluids, but also it can produce ripple and backflow due again to the mechanical force that is exercised on the tubing in order to push the liquid. And this case too, like you will have a lot of oscillations in the system because of this mechanical force. And the third option that we adopt here at Fluigent is the pressure controllers, where we can apply a pneumatic pressure producing smooth, continuous flow with fast response time, as there are no mechanical force based on this technology. So basically, the difference of pressure between the reservoir, the pressurized reservoirs, and the external pressure that will generate the pushing of the liquid from the reservoirs towards the sample or the chip. And in this case, we limit the oscillations because like there are no mechanical force in the system. So briefly, at Fluigent, we develop a pressure controller system that are specifically designed for microfluidics. By regulating pressure instead of displacement, we achieve like high stable pulse-free flows, as you can see in the graph, compared to a classical syringe pump with rapid response times. This approach is ideal for delicate biological experiments, including organ-on-chip, droplets, and long-term perfusion. What we'll discuss today and what will be a plus is that the pressure-based flow controllers now, you can also integrate them easily with flow sensor, and it can be a way into introducing automation through the experiment by dedicated softwares or the classical coding protocols that are on the market or in the field, ensuring like precise monitoring and easily handling compared to the classical methods. So before starting, I would like to present like the classical microfluidic setup that we have, that we typically use. So basically, it consists of a pressure source connected to a flow controller or to a pressure controller, which pressurize a microfluidic reservoir connected to a flow sensor. And it can be an optional valve if you are looking for more practicality towards the microfluidic chip. Classically, the microfluidic chip is observed under the microscope to see what's happening inside. And overall, all the setup can be controlled via software that can be dedicated for the flow, for the pressure, and control the overall system of the experiment. However, in order to control and to have this response time, and as you can imagine, the flow is essential. A stable flow is essential, particularly for experiment involving the living cells or biomedical applications. Our technology is based on a direct flow control, or a DFC algorithm, which continuously monitors flow via sensors, and it can adjust pressure in real time. This maintains a constant flow rate and a uniform, for example, shear stress or the flow across the microfluidic channels with high response time between the communication between the flow sensor and the pressure controller in order to increase or decrease the pressure in function of the variations that you will have in the chip. And of course, in order to maintain or to satisfy the specifications that you ordered or that you are doing your experiment. Based on this algorithm, we developed this oxygen software, which provides a unified interface for controlled pressure, flow, and the valves that we use. It will allow you to design sequences, ramps, and time-based protocol while logging data for the reproducibility. By automating these processes, the software ensures stable experiment conditions over a short or a long duration, reduces the operator errors, and support complex workflows in the system. So basically, using the oxygen, you have also the option of creating your own protocol and adding loops, weight, and so on in order to streamline your experiment or your microfluidic specifications. In order to dive into these approaches, we will discuss in the next slides 3 case studies. The first one, organ on chip, the second, droplets in microfluidics, and the last one dedicated to Omics application, where we can see how we can introduce some automation towards your microfluidics experiments. So for the first case study, I'm pleased to be joined with our customer support engineer, Maya, who will explain to us the first case study, specifically the recirculation for organ-on-chip applications.
Thanks a lot, Joseph, for the introduction. And yes, so I will focus more on the recirculation part today and explain especially why the precise flow control is crucial in this type of experiments and how we can automatize it. So today we know that there is a strong need to run biological tests using models that better mimic the human in vivo conditions. And we now have several alternatives to the classic 2D static cultures, like for example the 3D cultures, and more recently the organ-on-chip models. Which offer a much higher level of control over a lot of biological parameters. So to recreate the human in vivo conditions, organ-on-chip systems must provide tissues with a flow that is stable, continuous, and physiologically relevant. In the body, there are a lot of circulating fluids, such as the blood, the lymph, or the interstitial fluid. And they have several roles in the body, like delivering nutrients or removing the waste, but they are also applying mechanical forces to the cells, and that can guide their behavior. So to mimic this environment in vitro, it's very important to have a very precise control of this flow. This is especially important for active tissues that we often have in organ-on-chip models, like liver, kidney, or gut, intestinal epithelium, for example. Indeed, if the flow is too low, then you can have a gradient of nutrients or oxygen that can appear inside the channels, and this can lead to hypoxia, metabolic stress, or even cell apoptosis. And on the other side, if the flow is too high, then the cell can detach or become overstressed and mechanically overstressed. So that's why only an appropriate flow rate supports the long-term homeostasis across long-term experiments that we need with the organ-on-chips models. The shear stress, especially, is one of the key points that the cells are responding to. Indeed, like cardiomyocytes, many stem cells, they adjust, for example, their morphology and their function based on the shear. When the shear stress is correct, the cells align, as we can see, for example, in the scheme. But also they polarize, they form tight junctions, they adopt certain gene expression, and they have like generally a good, a healthy metabolic activity. But if the shear becomes unstable, for example, if there are spikes or pulsations in the flow, then it can trigger to inflammation, break cell adhesion, or change the cell phenotype. And also for organ-on-chip, we are working like in microchips. So, and because microfluidic channels are extremely small, even small variations in the flow can completely modify the shear stress. That's why solutions like manual pipetting or pump with pulsation that introduces oscillation inside the flow, the cell can interpret it as an abnormal mechanical stress, and for sensitive stress, we have to avoid that. So yeah, in conclusion, like long-term organ-on-chip cultures relies on this stability, and this is why the choice of the pressure controller is very important for this kind of experiments. So different technologies exist. Here we are comparing two main technologies, the peristaltic pump and the pressure-based flow controller. So the different technology all have their advantages and disadvantages. For the peristaltic pump, it's very like commonly used, so they are quite straightforward and easy to use, but they are also coming with some limitations, because like the rolling mechanism of it produce some pulsations, and also the tubings can damage over time, and the risk of contamination is higher. You can see here on the scheme that there are pulsations on the flow. In addition, the shear stress generated by peristaltic pump can be too high for sensitive cells. That's why for this type of application with sensitive cells, now pressure-based flow controller is like I would say the best solution, because it provides more precise and stable flow regulation over time. Using these flow controllers, you can build setups that are perfectly adapted for recirculation. So one example is the setup that you can see here with the recirculation package. And so it's really a flexible setup, pack with everything, and that integrates perfectly with basically every organ-on-chip experiment. So to enable recirculation with this setup, the system uses two pressure controllers, so two pressure lines that are connected to two reservoirs. It also included a pair of switching valves and a flow sensor. The flow sensor, it ensures the consistent volume exchange and keep the shear stress very controlled and low for this experiment. The idea is quite simple. Like the first fluidity apply pressure to the first reservoirs, and that pushes the medium through the system, so through the valve, and then through the microfluidic chip. Then it goes through the second valve and goes to the second reservoir and filling it. After a defined volume has been transferred, then the system can be programmed to automatically switch it, and then the pressure is then applied to the second reservoir, and the valve switches the position, And so the liquid goes from the second reservoir to fill again the first reservoir. And so you can automatize it to do it for weeks, days, how long do you want. And what's very important is that inside the ship, the flow remains unidirectional. So to automate this type of experiments with the setup that I just show you, we can use the oxygen software, which is a Fluigent control software. So here you can see an example of setup, an example of protocol that was made with the setup that I just described before. So the goal here is to run a recirculation at a very stable flow rate of 500 microliters per minute. So you only have to enter the desired flow rate in oxygen, and then the pressure will automatically adjust to reach and maintain that flow rate. Then we use the function wait for volume to control exactly how much medium has passed through the system and to switch when we want to make the recirculation. In this case, once 1,000 microliters have been transferred, then the recirculation switches direction. And so you can clearly see here the switching of the valves, as I'm showing you, and to fill the other reservoir. So this type of automation is ideal for long-term studies that need stable, hands-free. Protocol can be run directly on oxygen, but it can also be integrated in other software environments. For that, Fluigent provides a software development kit, so the SDK, that allows you to code the protocol in different languages, such as Python, MATLAB, or C++, for example. With the SDK, you can create very complex protocol with loops, dynamic adjustments, and so on. Here, you can see an example of code that was made with Python, and it's exactly the same protocol that I just showed you before in Oxygen, but here written in Python. So the code is organized in 3 main parts. First, you have the import and initialization section. This is where we load all the Fluigent libraries, connect to the Fluigent devices, and all defining all the initial parameters, such as the flow rate, the pressure range, and so on. In second, we defined the functions. For example, here is the definition of the function wait for volume that I just explained before. And in third, you have like the device control, the recirculation protocol itself. So this part controls the sequence. It runs the flow, runs the wait for volume that you already defined, switches the valves, and so on. And you can repeat this cycle as many times as needed. So using this SDK makes the experiments fully scriptable and perfectly reproducible, and especially if you want to integrate it into another software. We also provide a lab view control option, which is especially useful for engineers and automation specialists. So here is an example on how you can use it. You simply can have a look at your Fluigent folder, and you have different folders with different types of setup with different products and systems. Yeah, then you can see, yeah, on live view, you can also create and change your setup. You have different functions that you can add, different, of course, the different products, and link everything all together with your other products you have. And in this example, you can see a program pressure ramp, so pressure steps with the fluidity. So you can see in black, it's the order asked, the pressure order asked, and in orange red is the actual pressure in the system. So now that we have seen how recirculation can be automated using Oxygen or even coded with the SDK and different languages, I'd like to present you another solution that is aimed for recirculation and make some comparison between both. So on one side, we have the traditional recirculation pack that I already presented. Basically, it's a classic microfluidic setup with all its tubing, different parts and fittings. And you can see everything that's inside. And on the other side, you have the OMI, which is a fully integrated solution designed specifically for an organ-on-chip solution. So in fact, it's like if the setup on the right is integrated inside the device. So the recirculation pack, its advantages is that it's very customizable, modular. Like you can choose the tubing, the valves, you can choose the reservoir, like everything that you want. And also you can add pressure modules, you can change your setup if needed, and so on. But it requires some space, manual assembly, and I think also a certain level of microfluidics expertise to use it. And on the other side, the OMI is so all integrated. It's very compact. You can put it easily in the incubator or like on this picture under the microscope to see. So really easily to transport. You even have the pressure source that's integrated inside, and you have a lot of internal routines that are like pre-engineered. It also comes with the tablet and with a specific software when you can automate all your setup. So, which means much faster setup, and compared to the other setup, far less tubings and fittings and so on. Here it's a short video showing the OMI so that you can see a bit the device, its size, and how to use it quickly. So as you can see, it's really compact. It's composed of 4 reservoirs. You can put it just inside the incubator, take it back, and have a look inside your microscope of the results. It's really easy to use, like everything is straightforward. You don't need any microfluidics expertise. And basically, you just have to connect your chips to OMI, make your protocol, and that's it. And it's still reliable because you have the same technology inside. Talking about technologies, you can see here like all the components that are inside the OMI. So you have an integrated micropump for the pressure source, a liquid level sensor inside the reservoir, a disposable cartridge where you can put your solutions and change it for the sterilization, and a touchscreen interface to control everything, the chip holder, 4 reservoirs, and yeah, that's basically it. And of course, a flow sensor inside to monitor the flow. OMI allows you to monitor 4 main functions, and you can see the corresponding path on the right with the yellow path. First, the first function is the perfusion, so the medium flows from one reservoir to another and providing a continuous supply of nutrients and so on to the cells. In second, you have the most commonly used, which is the recirculation. Basically, it's the same as the perfusion, just there is a refill step between the 2 reservoirs so that you can do it during weeks, days, and so on. So you have a closed loop, you're reusing the medium, and you can also control the flow. The third is the injection. So it's used if you want to inject drugs, for example, or any other reagent at a timed delivery. And finally, the last function is the sampling, which enables automated collection of small volume for metabolics or other omics analysis. Here is a screenshot of the OMI application on the tablet. It's really user-friendly to create the protocols. So the apps, it's built in blocks, and you can choose the different functions that you want. For example, here you have recirculation, injection, and then recirculation sampling, like the function that I presented just before. And also you have ready-to-use protocols that are included, such as the calibration, the sterilization, the loading at the beginning of the experiments, and then the cleaning. So everything is ready to start your experiments. And here you can see an example of a recirculation protocol running in OMI. So here is the recirculation. So the medium flows from one reservoir to the other and go, of course, through the chip. And when the first reservoir becomes empty, OMI automatically detects it. And then at this moment, the system performs a refill between the two reservoirs. And that's exactly what you can see here on the interface. Of course, during the experiments, you can monitor pressure and flow rate in real time. And if you automated everything, at the end of the experiments, you can, of course, download and save all the data. So you will get a graph like this with the pressure along time and the flow rate along time. So the pressure in purple and the flow rate in orange. And here there is a zoom so that you can clearly see like the refill phase. It's approximately from 1 minute, and you can see that the flow rate is very stable during the experiments. So now that we've seen how to automate recirculation experiments with pressure-based flow controllers, let's move on the second case study with Joseph, where we will explore how to automatize the microfluidic droplets experiments.
Yeah. Thank you, Maya, for the two approaches from the recirculation pack and the OMI approach. So don't hesitate if you have any questions about this first part to add them in the Q&A, and we'll answer them by the end of the session. So now we will move to the case study 2, and that's why like one of the advantages of microfluidics that you can work in several fields across while talking the same about the precision, the advantages of the microfluidics. So for the case study 2, we'll focus on the droplets in microfluidics, and particularly how to produce them compared to the classical methods that we use. So in microfluidics, droplets are generated with precise control over size, frequency, and uniformity, enabling high reproducible microreactors, in contrast to the traditional batch that you can see, for example, in the images, either the technique or the microscopic images of the obtained droplets, where it produces heterogeneous droplets with broad size distribution. One of the important parameters that we are talking today about is the stable flow rates, which are essential for controlling droplet monodispersity and ensuring reliable experiment outcomes. So even though like each method has its advantages, for example, for let's say for the mixing, you have rapid emulsification suitable for various formulations, and it can be easy to scale up for bulk production, but it comes with challenges such as the broad droplet size, the heat and shear, which may degrade the sensitive materials that you have, and it can be difficult to produce more complex or structured emulsion compared to microfluidics. So that's why at Fluigent, we combined our flow control expertise with Sequoia's droplet generation offer, represented by the ray drop, in order like to create single or double emulsion via a controlled way. So that's why we have developed a microfluidic path through the platform, where we combine the flow easies that we presented earlier with the Sequoia's ray drop, in order like to have simple or double emulsion with a high reproducibility and monodispersity of the droplets that we are creating. So how it is designed. So again, like you will have the link in order to connect to the software or to your PC system. And afterwards, like we'll have 3 lines of the Fluidics. Each line is connected to a pressure reservoir, where it is one for the continuous phase, shell phase, core phase. And each one can be connected to the adequate fitting of the ray drop, either from the chamber in order to do the continuous phase, the shell, or the core phase. You may ask, for example, why we can have 2 reservoirs per line. So it's for practicality in order to help to do one, the priming and the cleaning of the system, and the other one where you can load your sample. We can add some switches or valves in order to make the experiment more smooth in terms of passing from one protocol to another, such as the cleaning and the washing, the priming, and so on. And also we combine it with the fast camera in order like to see in real time what's happening inside of the microfluidic chip and tune the system. What is advantages and what can be an advantage in microfluidic and the droplet field is that you can really tune the parameters that can affect the sizes of your droplets. So for example, here in single emulsion, you can see the continuous phase inside of the chamber and the droplet phase, the monodispersed phase that will create these monodispersed droplets. In fact, here, like you can unlock some parameters, which is not possible through mixing or the classical methods, as for example, the effect of the changing the flow rates compared to the size. So for example, here we can consider it, or we studied the variation of droplet phase flow rate and the continuous phase flow rate. And we concluded that, for example, if the continuous phase, the flow rate of the continuous phase is different, you can change the size in a really predicted way, the size of your droplets, as you can see in this graph. The same goes with the double emulsion, where here more precision is required in order like to tune the core, the shell, and the outer phase. Again, like we can see by tuning the flow rates, you can achieve like thin droplets, including the shell, or more thicker shells compared to the first one. So here again, for example, if you vary the shell phase, you can have bigger or wider droplets, shell droplets. And of course, too, as we saw, if we change the continuous phase, it can affect the droplets. So in this case, that's one of the advantages, like when we pass to microfluidics, and it can be a way in order to control it in a really automated way, rather than switching manually each step in order like to reach the best configuration of your droplets. So basically, also through oxygen, we can either tune with pressure or with the flow rates. In order like to create or like to keep the dispersity of the droplets that we are creating, and even with the constant frequency throughout the experiments. And that's why one of the advantages, for example, if like there is some clogging, some issues, the pressure will adapt itself in order to keep the flow rate. Here, for example, around 315 for the continuous phase. The same goes for the shell or the core. The pressure, again, through the algorithm between the flow sensor and the pressure controller, it will adapt itself in order to maintain this stability in terms of the flow rates and of each phase, and thus the distribution of your droplets or double emulsion droplets. Based on this technology, we had several proof of concept that you can find it on our site. Basically from single emulsion, if you are interested in creating droplets or beads, such as alginate or PLGA in various sizes that can go down to 10 micrometer, for example. For the microcapsules, if you are interested into double emulsion, you can also do the polymeric microcapsules such as PLGA, chitosan, PMMA, PEGDA, or multiple emulsion. It's also possible using the ray drop and the Fluigent. And also it can be a possibility if you are interested in doing encapsulation of biological material, such as, for example, bacteria, yeast, and integrate them into afterwards, for example, to do some fact sorting throughout this experiment. So all these proof of concept are available, and you can download them directly in order like to have, if you have any questions or if you would like to integrate them in your microfluidic experiments. And finally, now we will move to this quickly for the last case study about automated perfusion approach for spatial omics, which also a field which can gain a lot using the advantages of microfluidics. So basically, the traditional immunostaining workflows rely on manual pipetting to add antibodies, for example, wash buffers, and other reagents. While it is functional, this approach introduces significant variability, such as the reagent coverage can be uneven, for example, which can lead to inconsistent staining. Secondly, the timing difference between manual step, it can affect the reproducibility. The mechanical disturbance can damage the delicate cell layers, especially as we saw earlier with the simulation of the pipette tip, which can damage the cell layers, the tissue construct, or the microfluidic cultures. And also the manual handling is particularly challenging in long-term or multi-cycle staining protocols, where multiple rounds of washing or intervention is needed to do the imaging that we would like. So one of the solution, also a plus to the Fluigent that we suggest, is the area, which is also based on the pressure control that the pressure-driven system that we provide, but it is dedicated to microscopy and these types of application. So basically, like we'll have 8 reservoirs for the samples, in addition to 2 others for the washing. And afterwards, by automating with the dedicated software, you can add chronologically the steps that you require and the time and all the protocols that is needed throughout the system, and you will let it run directly without any intervention compared to the classical pipetting methods. So to demonstrate this, we'd like just to have like an idea about the method that is typically used. So for example, you can have for this application, so the area dedicated software can provide a step-by-step guidance and allow you to define the internal workflow in a single protocol. So for example, here, the protocol that we'd like to do is to inject 100 microliter, incubate, wash with for 10 minutes, inject, and afterwards incubate again for 1 hour. So here, how we can, let's say, stream or control the flow using the system. So using the area software, we will do these steps. We will program these steps. So the first one, it will be, for example, volume injection for 100 microliter per minute, wait for the incubation for 30 minutes, and afterwards plan again another injection. You can work either by flow rate or by volume. So it's also it's a possibility depending on the system that you provide. And finally, do a wait for 30 minutes. In this case, we can simulate, or like you can have an idea about how we can replicate typical protocol into the area software and do the experiments afterwards. The area system, like it can be connected or added to a 2 switch if you are interested in only 1 microfluidic chip and 1 sampling chip. But also it can be provided with the M switch, where we can increase the number of connected chips up to 9 samples compared to the system that you will have. So the 2 version can be a possibility using the same system. One of the also advantages using the system that the area can be integrated with microscope and imaging system using TTL or digital triggers. So this synchronization can allow real-time reagent delivery coordinated with imaging events. So the microscope and the area can communicate with each other in order like to have or exchange the informations and steps. Control perfusion during time-lapse experiments. And it can automate execution of complex protocols that require precise timing between the staining and imaging. As an example of or more elaborated protocol using the system, where like, for example, you can see here the accumulation of volume injections and also the potential, the possibility of waiting for user for its confirmation to pass from one experiment, from one step to another. But also here, like you can see the wait for TTL in order, for example, if the area will receive a signal from the microscope in order to pass to another step. So all these possibilities can be suitable using the same software that we have. Here is just an example of imaging that we had based on the IBEX protocol, which stands for the iterative bleaching extend multiplexity, and it is which is a high content multi-cycle immunostaining workflows. So we have several papers or proof of concept using this and application notes on the site. Also, like you can, if you are interested in these types of application, don't hesitate to reach out or like to check the papers available on our site with this. So finally, to conclude between here, like it's a schema in order like to have a clearer idea about the comparison between the pipettes and the automated protocol from the limitation of the pipetting, time consuming, uneven fluid delivery, the errors and the solution mismatches compared to the advantages in terms of time saving, steady fluid delivery, sequential distribution, and higher reproducibility of your system. So with all these applications, we hope that you had a better idea about how we can include some automation in your system using the microfluidic experiments that you have. And we can now open for your question if you have any particular questions throughout the 3 case studies or the introduction also. So please don't hesitate to add them in the Q&A panel in order to discuss further. In the meantime, I will present briefly Fluigent as a company. So we are based in Paris with a subsidiary in the US, in Boston. And we have a wide network of distributors in Asia and Australia. So with our main products, the Flow-Easy and the other products that we showcased today interfere or they are present in prestigious papers throughout the journals that we have and with several collaborations with wide universities across the world. So don't hesitate to contact us if you have any upcoming projects or you would like to include or optimize your microfluidic setup and be based on pressure using our system. So with this, I would like to thank you again for your participation today to this webinar. And stay tuned for the upcoming ones with the Fluigent. Thank you for your time. Have a nice day.
Have a nice day.